All right. Good evening, everyone. Good evening. Good evening. My name is Michael Schatz. I'm a professor at Johns Hopkins. I'm also one of the PIs for the Galaxy Project and also part of the organizing committee for GBCC. So first of all, welcome to Cold Spring Harbor Labs. I'm really excited for this event. It's been, I don't know, more than a year in the planning and in the making, and it's just great to see so many people here. This is going to be a great week of science, of discussions, and just really lifting us all up in this time right here. Woohoo! So one thing I wanted to ask, getting started here, how many people here is this their first time ever to Cold Spring Harbor? Yeah, that's kind of what I thought. I thought this would be kind of a new experience for many people. I was actually on the faculty here from 2010 to 2016, so this is kind of a homecoming for me. And there's just a long history of science that has happened here for both the Galaxy, the community, and also the Bioconductor community. So we thought that this would be a great place to come back for this year's conference. So it's a really interesting lab. It was founded in the 1800s as an agricultural research station and has been home to many, many, many discoveries and many, many presentations about molecular biology and genomics and just sort of science and beyond. There's a beautiful campus here, and we're really fortunate to have a great week of weather, although extremely hot. So make sure you're drinking tons of fluids and staying cool. But I really encourage you to check out the campus. So this is the so-called lower campus. This is where the conference center is. As you kind of wind your way through the campus, this is actually the research labs. There's like actual hardcore molecular biology happening in these little cottages. And part of that experience, if you want to check it out, we do have organized this year a little scavenger hunt. So I hope you're all on the GBCC Slack. Hopefully you got an invite there. There's a scavenger hunt channel where there's, I think it's 11 little puzzles to solve. And then the idea is take a picture of it. It could be you as an individual in a small team. And then if you submit your pictures, you can potentially win some prizes. But really, it's just an opportunity to kind of explore the campus. There's just a lot of beautiful history and science and culture that has been celebrated here. So as I mentioned, Cold Spring Harbor has been home to many remarkable discoveries. So there is a small research facility here. There's about 50 investigators, a few hundred PhD level scientists. Here's just a few of the most notable scientists here, Nobel laureates over the years, starting with Barbara McClintock, discovering transposons, AKA jumping genes. Alfred Hershey sort of designing and carrying out the experiments that proved that DNA was the genetic material of life. Rich Roberts I think it was in the 80s, discovered that there's this very obscure thing called splicing that occurs all the time and is really important for the formation of proteins through the central dogma. A recent Nobel laureate was Carol Greider, which she did a lot of her work here looking at the activity of telomeres. Another kind of great tradition of Cold Spring Harbor is this great tradition of quantitative biology. Going back to 1933 was the first annual symposium. And this is a tradition that carries on to this year. So we're coming up on 100 years of quantitative biology here at Cold Spring Harbor. And again, it's just sort of year after year. What are the latest and greatest techniques? What are the analysis techniques? How can we make those discoveries given sort of very rough and very noisy data? That's been a really important part of biology for the last 100 plus years. Here's another really important figure at Cold Spring Harbor. Does anyone know who this is? This little pop quiz. Claude Shannon. So why would Claude Shannon be notable here? Shannon entropy, the father of information theory. So Claude Shannon completed his PhD at MIT. But one remarkable thing is for his thesis work, and this is an actual picker, his thesis was on an algebra for theoretical genetics. He actually did that research here at Cold Spring Harbor. So look to your left, look to your right. The next Claude Shannon could be sitting right next to you, inventing something as important as information theory and entropy. That could be right here. So this campus, as I mentioned, has been a really important part of the Galaxy tradition and also the Bioconductor community for many years. So the Galaxy community just had a really important milestone. So the first commit to GitHub was on June 1st, 2005. Actually, there's a little bit of archaeology at play, but that's the year, that's the date that we're celebrating there. So it went from, just setting up the basic repo and getting things organized. And here we are 20 something years later, and we have this really mature platform. This is a screenshot I took last night. We just got our latest release out with amazing features. I think we can all be really proud of this work. Part of this as well is the very first developer conference was here in Cold Spring Harbor 15 years ago. So we thought it was time to come back. And those conferences has just been a really important part to the Galaxy community over the years, as a way to kind of strengthen ties and talk about technical details and share the latest science and talk about the future and maybe have a beer or two together. You'll notice that was prominently part of the schedule from that original meeting. So that's a little bit about the Galaxy project. And as we're planning out this year, we're like, well, we've had a great 15-year run of this. But increasingly, we've been working closely, more and more closely with different communities. At the top of the list has been Bioconductor. There's been a number of really important projects. In my world, I'm very active in something called Anvil, and we have, I don't know, weekly or daily calls, it feels like, where we talk about how can we align on advancing on human genetics, but then kind of more broadly, how do we advance on data science? This really, really close partnership between Galaxy and Bioconductor. So that just sort of brought us together, I think, in this great community. And by design, you'll notice the program is very much interleaved, right? We didn't want it to have like parallel conferences. What we wanted to do is kind of bring people together. We're all here in support of open science. We're all here in support of really rigorous science, reproducible science, accessible science. There's all these really great core principles. And it's just really important to us to kind of bring everyone together. If I could still invite up for just a second here, Anton Urchenko.
I am. I'm Anton. So we really wanted to make this conference intertwined between Galaxy and Bioconductor because, of course, we have a lot of Bioconductor packages in Galaxy. We have a lot of tools based on Bioconductor packages, but it just makes sense. And the reason why it makes sense because of this tree. So, well, it's a tree. If you think about any biomedical analysis, of biomedical data, it's sort of like a tree. It usually starts with some QC. Well, there are well-defined tools for that. Then you kind of branch. I don't know, maybe you're doing RNA-seq, maybe you're doing variant analysis, maybe you're doing some epigenetics. But it's sort of all kind of, progresses along well-defined branches. And then at some point, when you get sort of to the leaves usually are what? I don't know, some tab delimited data sets that you need to actually interpret to publish. You don't publish tab delimited data sets. And so historically, well, how does that work? So historically, Galaxy was kind of very good with doing things which are well-defined because you can combine them into workflows. And because Galaxy is supported by very powerful free compute infrastructure. But once you get actually from whatever you have your reads, you get them into, I don't know, variant lists. But again, you don't publish variant lists. You need to interpret them somehow. And that's what you use Bioconductor for. So it's a really kind of a partnership which two projects are very congruent to each other. You can do big, heavy stuff with Galaxy workflows, and then eventually kind of transit to publication figures eventually using Bioconductor packages. So I think the two things fit together very, very nicely. And one of the goals of this conference is to inform Bioconductor people what Galaxy can do and inform Galaxy people what Bioconductor can do. And by doing this, I think we can strengthen both communities and perhaps become one.
So we're really excited about this. Again, we think there's great opportunities to work together. So by design, we're trying to interleave and sort of connect as much as possible. Just very quickly, I want to say a few words about the schedule. So tonight, we just have the sort of opening remarks. After myself, we'll hear from Vince Carey just to hear a little bit about what's new in the Bioconductor world. And then we'll have kind of a quick round out on what's new in the Galaxy world. Tomorrow, we have a really exciting program. We're going to kick it off with a keynote speaker, Sergey Pond. He's a professor at Temple University, talking about interactive genomic analysis with responsive notebooks in the browser computation and AI-assisted development. Throughout the day after that, we'll have other sort of abstract selected talks. We'll have a bunch of discussions and trainings and oral sessions. In the evening, we'll have our first poster session as well. Then on Wednesday, we have another really action-packed day. We'll have our second keynote speaker, Charlotte Sonneson. She's at the Frederick Meiser Institute for Biomedical Research, talking about from classes to the community, how Bioconductor has advanced my research. A couple other sort of special events on Wednesday. So one of them will be lightning talks. So those of you that are interested to kind of come up stage here and just say a little bit about your work, typically, this will be sort of centered on what you're presenting for your poster, although it's sort of up to you. You'll be given an opportunity to have up to 4 minutes, and we will get a giant hook at the 4-minute mark to kind of escort you off stage here. You can have up to 5 slides. I would actually encourage you to have 1 slide. I think that for a lightning talk, 1 actually works better, but if you want to have a couple animations or whatnot, you're allowed. I would not get up and give the micro machine version of just giving your talk really fast. I would discourage you from that. Think of this as like a brochure, highlights, just a little advertisement. And then the idea is if people are interested and want to learn more, they can track you down at the conference and have kind of follow-up discussions. So there's a bit.ly link, there's a QR code, there's an email that went out on the Slack as well. all the information about how you can sort of register for lightning talks. It's first come, first serve. So we're excited to do this. This is always a highlight of a conference, but we really want you to sign up for this. Another special thing is Wednesday evening is the infamous or famous Cold Spring Harbor Lobster Banquet. So I encourage you to come check that out. Have some delicious lobster. We got Michelle here. We got Natalie here. Another sort of special thing, because it's our 20th anniversary of Galaxy, we've kind of organized a little celebration. Although it's open for everyone, please come. We want everyone to participate. And then tradition would have it very late, often at like midnight, 1 a.m. or even later. There's a great late night pizza place. So if you're interested in late night pizza, there's something that is really weird but delicious called cold cheese pizza. You gotta try it. You tell all your friends. It's weird. It's a Long Island thing, but it's delicious, and especially at 2 a.m. You definitely gotta check it out. Okay, and then on the last night, Thursday, we're going to close it out with some more discussions. We'll have our third keynote speaker, Jason Williams, from here at Cold Spring Harbor. He's been a friend to both Bioconductor and Galaxy for many years, presenting on conquest of abundance, genomics in a time of plenty. Immediately after the conference here, some of us, if you signed up for it, if you're interested, we're having a sort of a partner meeting in New York City, right in Manhattan, at something called CoFest or Collaboration Festival, where it's going to be really hands-on. If you want to get, do a bunch of hacking, if you want to get sort of started on, I don't know, tinkering with the internals of Galaxy, tinkering with the internals of Bioconductor, we got a bunch of different groups there that will sort of help you get onboarded. And, if you want to sort of speak and work directly with the developers. So this is very much a collaborative festival. So we have another place, another QR code where you can sign up and sort of present your ideas of what you'd like to work on. It's a 2-day event. So you're probably not going to write a whole, I don't know, new Unix kernel from scratch. But if you want to kind of get started, or if there's a project that you want to get, kind of have some focused energy on it, this would be a great time to sign up and work on this. So this is hosted at Weill Cornell Medical School in New York City. I think registration officially closed, but no. Oh, Jen's giving me, if you're still interested, we can certainly accommodate you. We have plenty of space for it. I will caution, though, it'll be up to you to find your own housing in New York City. We do have a few leads and pointers, but it's a little bit too far to sort of commute from Cold Spring Harbor into New York City. Okay, next up, I'm going to invite up Michelle Savage, who's going to say a few words about the Code of Conduct. She's our sort of liaison for both Galaxy and Bioconductor on the Code of Conduct committee.
So this is just a quick reminder that when we register for the conference, we all agree to the Cold Spring Harbor Code of Conduct, which is pretty comprehensive and actually overlaps a great deal with Galaxy's Code of Conduct and Bioconductor's Code of Conduct. So nothing unfamiliar there. The one notable exception is they have some limitations or restrictions on alcohol use on campus, limiting to some certain areas like Blackford Bar. And I want to say during events like the wine and cheese and dinner and stuff like that. So just keep that in mind. Discretion is always good. As a representative Galaxy, we at our annual conferences, we also report on the number of Code of Conduct violation incidents. And while we have had issues in the distant past, I'm happy to say we haven't had any in the last 12 months. So thank you so much for participating in that. And then just a final note that if you do need, if something does happen or we witness something, please feel free, please do report it. I think Cold Spring Harbor has a mandatory reporting policy. Those are the people, but also to share it alongside. Also, you can look for people like me if you need help or support with that and some of the other people on these teams. That's it. Thanks.
Yeah, and we're here to have fun and to share. We hope that everything's going to go super smoothly. Right, we haven't had any issues. But again, we wanted you to know that if you have questions, you have concerns, please reach out to any of the organizers, Michelle, the staff here at Cold Spring Harbor. We're here to support you and really support you. Okay, for these intros, there's a bunch of people we wanted to thank that really made this conference possible. There was a very extensive sort of committee of organizers to help sort of set up the logistics of the program and how we're going to do the training and how we're going to set up the talks and the presentations and so forth. There's dozens of people here, and I thank all of them. If I had to call out two that were really special, the scientific chairs, Ennis and Jenny, thank you very much for all your hard work. I know it's been a year of conference calls to kind of set up the whole program. Thank you for with setting up this excellent program that we have here. In addition to them, I'd really like to thank all the sponsors that really supported a lot of people to attend here. It kept the costs down for a lot of people. It also provided support for individuals to travel to this conference. So we had 2 silver sponsors, the Chan Zuckerberg Initiative and Bristol Myers Squibb. We had 3 bronze sponsors, Limigrain, GalaxyWorks, and the ARC Consortium. We had a few friends of GBCC, that's GigaScience and Elixir. And then we also had a conference scholarship program that was funded by NHGRI, and we're really grateful to all these programs for the support that they have. In addition to these groups, ever since the passing of James Taylor, we established a non-for-profit called the JXTX Foundation. It's a foundation for open science, really designed to help support junior scientists launch out into their career. James had this really special quote, right? The most important job of senior faculty is to mentor junior faculty and students. And I can really just say that this is a big part of our identity and our commitment is to making sure that this legacy carries forward. And we're really proud to say that in support or in sort of honor of James and all the work that he did in Galaxy and beyond, at this meeting, we were able to support 6 different junior scientists attend the meeting. So we have Sudipta, Grace, Negan, Marella, Nitin, and Arish. Are we in the room here? Any chance? Okay, people are trickling. Okay, so congratulations to all of you. I will say this was a very competitive process. In addition to kind of all your science, we asked you to upload like your GitHub repo. This is like one of those special events when your GitHub history matters a lot more than your Google Scholar history. We really are looking for commitment to open science and what sort of contributions that you're making. In addition to these 6 awardees, we also had a separate travel fellowship in honor of James Johnson. He was one of our great friends and mentors, especially through the Galaxy P project. And through that travel fellowship, we were able to support 2 additional junior scientists, Parikshish and Pawi. Are we here? Oh, welcome. Congratulations. And again, it was incredibly competitive. So congratulations to you. This is definitely something to list on your CV. And it's been fun. We've been offering these scholarships now for 5 years. And it's just been really remarkable to see those people advance in their careers. Some are in faculty lines now, and it's just been really incredible. So I encourage you to list that. Okay, so that was it for opening remarks. I'm now going to hand it over to Vince Carey to give us an update from the Bioconductor community. Thank you so much, everybody.
Well, Mike, someday you'll have to teach me how to make a slide. They're really wonderful. Let's see, this is slide 1. Welcome. We're in Cold Spring Harbor. How do I go to the next one? Green button? Green button. There we go. See? I don't know if you wanted to look at that, but that was my sketch. I guess that was in 2024 when I was very concerned about the idea that LaTeX would be going away. You don't know what I'm talking about? Sorry. Anyway, it was a very agitated moment for me. I might have been on a plane or something, and I decided to try and sketch what is going on in Bioconductor that nobody sees. The parallel devel release, the Docker binaries, the fact that we're using cloud things to get things on your desktop. Maybe we're on the desktop of most of the bioinformaticians in the world, and they're used to getting all these things very rapidly, very smoothly, and in a consistent way. And there are some innovations there, like, we started out back in 2002, 2003, GitHub didn't exist. So you have GitHub Actions there with a heart drawn around it, because I really want to make use of that and really get that going. Jetstream 2 also gets a heart. That's an academic cloud that's been very helpful to us in getting these things to scale and modernize, because we started out, with 20 packages, but then we had to keep going as people decided to contribute more and more, and the biotechnology diversified and so forth. So there's a lot there. And it takes a lot of energy for the folks that are involved, and I'll tell you a little more about what's going on. So we do get support from NHGRI, 17 plus years of funding. And it will close in March of 2026. We're hoping to get a renewal going. We've had a number of projects from CZI based on Bioconductor using GPUs, community support, tidiomics. We have a couple of folks that are key to that here. And then I was lucky to get one to work on ontology for Bioconductor packages. We're working with the EDM group at Pasteur Institute to improve the tagging of our software components so that people can find them in a systematic way. We do have a problem of sort of lots of projects using it, but we don't have any ability to capture any of the value that comes from their use of Bioconductor. And as funding in the United States scientific community seems to be in a special and difficult state, the idea of coordinating with other communities and with European investigators and investigators in Asia and so forth seems very important to keep the project going. And so this idea of a sustainability working group was floated to our TAB, our Technical Advisory Board, a couple of meetings ago. And I hope that if anyone has thoughts about that or would like to participate in that, they would get in touch with me. We used to use Slack to communicate with the community, and we could not afford that anymore. We hit Slack Pro. We've moved to Zulip, and I think that was a very smooth transition, thanks to work of Maria Doyle of University of Limerick. We released 3.21 in April, and Lori Kern took care of much of the review process there. She indicated here we had 7 to 8 active reviewers. How many folks here have submitted a Bioconductor package? So there are more that could do so. And if you do, you'll find that it's actually a pretty pleasant and informative process. So get on it. 2,341 software packages ranging from the year 2000 onward. We keep them running. 432 data experiment packages, 928 annotation, and so on. 5 books. I'll tell you a little more about those in a minute. The workshop instance, which I guess we'll be using here, I haven't paid a lot of attention to that. Alex Mahmoud is here, who has designed that and will be helping the teachers and their students to do the live coding that they want to do. And then we're also working on improving our CI/CD, and there's all kinds of things. We'll hear about WebAssembly tomorrow in another context, but if you want to use your Bioconductor packages in WebR in the browser, that can be done. Any questions, comments about things I've just said? Okay. We have some wonderful packages, mostly built by a longstanding member named Hervé Page. He couldn't be here now because he's traveling in France. The whole delayed array infrastructure, something called Genome Info DB, which gives you all the reference genomes for all the different organisms that you might want to work with, takes care of the naming of chromosomes and so forth. Our track layer, that's being worked on, again, also by Marcel Ramos, who is here. So there's a lot going on to just bring software to a more maintainable state and one which is more performant and has less vulnerability to collapse of web services or supporting infrastructure packages. We also have a lot going on in R, Python, in interoperability with a package called Basilisk. That is evolving to become more lightweight and to reduce the burden of bringing these mini Conda or mini Forge systems in every time you want to use something which is interfacing. I'll be talking tomorrow about these language agnostic data representations, which have to do with Alabaster and Takane. And then there are other issues which I'll tell you a little more about, sort of advancing our data technology. I'm a little concerned that we still don't see a lot of integration of large language models and other AI concepts in Bioconductor practice, but there's some movement afoot thanks to Michael Lawrence, who's been a long-time contributor. He's now at Genentech. Take a look at his GitHub repo called Wizard. It deals with a lot of the interfacing between R and LLMs for the sake of data analysis. We have a new stream of CI/CD, which takes packages that do use GPUs and tests them on GPUs. That was partly supported by CZI. And then there are governance activities with the Technical Advisory Board and Community Advisory Board. We'll have elections soon, and welcome folks to think about nominating. You can nominate yourselves if you want to be more engaged in these boards. I'll show you a little bit about a new approach to CI/CD. And then, as I've mentioned, there's all kinds of stuff going on at Genentech to help with data science and make it more, I would say, less lock-in to R and more usable with whatever computing infrastructure you want to try and use. So this is a view of some work that's been done by Marcel to bring the R-universe techniques into Bioconductor. I won't go into this in any depth, but have a look at R-universe and see how it conceptualizes a universe of software for the sake of developers and users. This is based on GitHub Actions and is highly scalable and could help move a lot of our activities from maintenance of the build system to other activities that are really needed. This is a view of a landing page in the R-universe framework. It gives the developer a lot more capability to design what their landing page looks like, as opposed to the current Bioconductor approach, which is a fixed format. Here's a thought about data. What we use, let's say, to ship out gene models and species-level genomics, we've been using SQLite for a good 15 years. And I think many of those packages are probably at least 5 times larger than they need to be to cope with the information. So SQLite is not really a winner for data technologies that we are using here. And you can see that we're also taking advantage of Parquet and DuckDB in other contexts. The numbers of downloads per month would sort of tell us, where should we put our energies in order to improve data technology here. And I think there's a lot of scope for work on this, but not a lot of bandwidth. Also in spatial transcriptomics, working with OME and GFF is an ongoing activity. If you want to learn about how to do modern statistics in biology or single-cell analysis or high-C analysis, there are monographs that are themselves subject to CI/CD. So you're not reading a book that the software doesn't actually work with. The software has been tested. And when there are problems, the developers, the authors are notified, and they can revise the book appropriately so that it continues to work. This is an example of the one for high-C. It's not just an authoring or distribution of published material. Jacques Sarizet also wrote a package called Make a BioC Book, something like BioC Book, I think is the name of the package. And all you do is set up some markdown in a certain set of folders, and then you compile into a monograph like this. And that's being used also in the spatial domain. Orchestrating spatial transcriptomics with Bioconductor is being done at BU with Lucas Weber. So that book is not yet in our system, but is finishing up and will be soon. How many times do people download packages like Sparse Array, which goes beyond R's capability for dealing with matrices that are very large? 500,000 downloads so far this year. There were 700,000 last year. Definitely worth knowing about. And here's a picture of our GPU stream. There are only 3 packages currently being tested, but there are plenty that use TensorFlow and Torch and so forth, and they should be moved into this system so that they get tested on GPUs as well. So a lot of detail. I think this just sort of summarizes what I've been talking about. The conferences and training have been a major part of Bioconductor for a decade, at least. And the workshop instance that Alex Mahmoud conceived of, which runs in a special instance of Galaxy, will be used in the workshops here. It addresses both the authoring of workshops and the consumption of workshops in a very elegant way. And I hope you will get a chance to use it and enjoy it. Last year, we had very good attendance, 648 workshops and package demos launched. 132 workshop platform users in one conference. So that's pretty impressive. And it all falls back to the commitment to training and the training committee, which is led by Charlotte Sonason, who is here, and you'll be hearing from in her keynote, connected with the carpentries, and I think a very, very potent training capability for building a genomics workforce in the world. Recently published a paper called 11 Quick Tips for Writing a Bioconductor Package, and that's in PLOS Computational Biology. So if you want to get on it, this will help you do it. And I'll close with just this blog post about Bioconductor in Africa, highlights from the first in-person course, and blog.bioconductor.org is certainly something you should be looking at on a regular basis. I even put a blog post in with a whole list of things that I plan to write about, which address many of the topics that I just talked about. So thank you so much for coming. And if there are any questions or comments, I'm happy to hear them.
Okay, so similar in spirit, let me invite up Anton. So we're going to tag team what we're calling here the Galactic Top 10. So Anton, why don't you just kick us off?
Well, what is it about? It's about galaxy, apparently. Okay, so well, so we just wanted to highlight some of the numbers from the last year. So sort of what are the 10 biggest things? Of course, so this conference in the US, and there are certain issues preventing people from other parts of the world traveling to the US these days. But Galaxy is not just in the US. There are kind of 4 major instances which are under this use Galaxy.star umbrella, which is pictured here. This is one in Europe, in Germany, .eu in Australia, .au here in the US, .org, and one in France, .fr. There are other instances coming up, one in Canada, in Belgium, and some of others that are listed here. And so this is, we call this Use Galaxy.star Alliance.
Okay, so that's number one. Number two has just been the explosion of usage of Galaxy. So this is a really nice dashboard. We're sort of connected to these major instances of Galaxy. We had a really major milestone in the last year, which is that we crossed more than 500,000 registered users on one of the different sites that are here. Very active community. We typically have about 10,000 users a month, new users a month signing into one of the different platforms.
Again, kind of, you know how to judge projects, how active they are. You look at GitHub, you look at the number of contributors. And I think both Bioconductor and Galaxy are very, very, very active in that area. And so the number of contributors for us is also growing, which is what we want to see exponentially, of course.
I should comment, this is just for the main repo for the main software. There's dozens more. Yeah, there's dozens more repos. One person that we really wanted to highlight, so there's a special group called the committers group that sort of oversee which pull requests are actually sort of appropriate and sort of validated. We have a new user that has just joined this in the last year, Ariza from the University of Feinberg in Germany. Unfortunately, he's not able to be here tonight, but we're really proud of all of his contributions.
Yes, and there is another reason he's not here because he was denied the visa. He's from Iran. So we have regular releases. The latest release was 25.0. It was managed by Ahmed, who is here, who is one of our recently new developers, but one of our star developers. He's involved into UI, for example, and he was involved in the release. And release process in Galaxy is kind of a very structured thing. We're aiming for, 2-3 releases per year.
And there's dozens of new features that were just announced that you'll hear a lot about at this conference over the next few days. One that I mean, it's very straightforward, but I'm really excited about is just to provide really simple visibility into a data set. So for a long time in your history, we have this expression, poke it in the eye in order to get a look at the data. It used to be just a pretty basic view, but now you can do really interactive visualizations immediately, just about making your data come alive and really accessible. All of this has been supported by a set of working groups that sort of manage different components of the software. And it's been awesome working with them. This is a global international effort. If I had to highlight one person that's, really helped organize everyone and keep everyone on track is Natalie Kucher, who I believe is here. Natalie, right here. So if you have questions about, contributing to Galaxy, and at a very technical level, come find any of us or come find Natalie. She's been amazing to help orchestrate this.
We work very closely with Bioconda and Conda Forge. So essentially, all the tools which are there can be easily incorporated into Galaxy, and current Galaxy instances have 10,000 plus tools. So essentially, all commonly used open source tools are already in any of those major instances, which I mentioned are under this usegalaxy.star umbrella. And wrapping tools into Galaxy is very easy. We're exploring some LLM approaches to that, of which you will hear at that conference.
In addition to the tools themselves, there's been a very focused effort on developing the workflows. We're going to see tonight an example of this through something called the Intergalactic Workflow Commission, which has been sort of hardening and sort of polishing up hundreds of workflows. Now, supporting this behind the scene, there's something like 10,000 tools. We don't have 10,000 individual contributors for that, but it's pretty close, right? There's a lot of sort of maintenance and overhead about doing this. And we're incredibly grateful to the whole community that has helped author all those adapters and make them integrated into Galaxy.
We're actively pursuing collaborations with large projects, and large projects come to us. So one of the examples I want to mention today is we are integral part, for example, of VGP, or Vertebrate Genome Project, where we essentially a platform which runs large scale assembly workflows. I think up to this point, we assembled something close to 250 genomes already. And if you know about this project, it's just finished at phase 1. And so we're going to have a number of publications coming in the fall. We have a new initiative called BRC. This relates to analysis of genomic data for pathogens, for different kinds of pathogens. We, of course, have very close relationship with Bioconda, because this is where vast majority of our tools comes. I don't know how many of you know that Galaxy is also a major part of biocontainer ecosystem. For example, all the Singularity containers that you pull from Galaxy resources. We're part of the Anvil project. You will hear about that during this conference. We have a strategic partnership with Elixir, with UCSC group, and this is also linked with BRC and so on. So there are many things which kind of hard to cover in one conference, but this what sort of strengthens our community, and our partnership with Bioconductor is part of this.
And then coming up in number 7 has just been sort of a huge thank you to all the members that have participated in the Galaxy Training Network. To date, we've, through the hard work of 490 different contributors, we've developed 477 tutorials. These are amazing, right? These are great resources that walk you through step by step how you can sort of execute an analysis, give you a little bit of background about what the different tools are doing, how to interpret results, how to QC it, how to diagnose different issues. There's great resources across a whole variety of genomics activities, but there's also great trainings and tutorials beyond genomics, into machine learning, into climate science, and beyond. Anything sort of data science, we probably have a training that's available for there. As part of that work, in this last year, we've had something called the Galaxy Training Academy. This is a worldwide effort to kind of introduce different people to the platform. And we're really proud to say in the last year, we had more than 3,700 registrants from around the world in 122 countries participate in this.
We try to develop this so-called communities of practice. So there are many communities of practice. Galaxy in itself has nothing particularly, hard-coded about biology. It's used in climate science, in computational chemistry, in climate research. And so here are some of the examples. Of course, these examples are more biologically driven, but there are more than that. And each of these communities has its own internal organization. They develop their own training materials. They participate in development of workflows and tools. And so if you are if you're doing something along those lines, I really encourage you to talk to us. We can help you to get into these organizations, into these communities.
Okay, another really exciting milestone in the last year is the sort of core Galaxy papers have been formally cited more than 20,000 times now. We know that this is way undercounting because a lot of people will just put in the URL or make a passing reference, but this is a formal citation to one of the major Galaxy platform papers. So these 20,000 include, some papers that we've written that we're really proud of, some work that we've done in the scientific community. But of course, the vast majority of these have been just from the larger community at large that have been contributing, building on, and using Galaxy to promote all the science that we're doing. And it's just been incredible to see this grow. And coming in for number 10.
Well, of course, this is the new world in terms of what we can do with AI and LLMs in particular. And I'm not going to reveal too much. You're going to see more of this today. I'm just going to say that we're working on this actively on various domains of that this brave new world.
Okay. And then, so that wraps up our top 10. I just wanted to again emphasize, so Wednesday night after the banquet, just in Bush Hall, there'll be a DJ, there'll be some, celebration about Galaxy, but more broadly, also Bioconductor, and then also open science as well. So everyone is very much welcome to come. We're gonna have some cake and some refreshments and some fun things as well. So if you're interested about anything we just saw here, it's really important that we stay in touch. So, pick your favorite platform. We've definitely moved into a multi-platform approach here where there's many different ways to kind of find us, talk to us, or just come talk to us in person. So that closes out our top 10. So we're now going to transition into a very new experiment that we're trying this year for the very first time, something called GBCC Live. So I'm going to invite up Ennis, Danon, Vince, Allison, Jeremy, and Marius to kind of kick us off now for a live demonstration of some of the latest and greatest technologies from both Galaxy and Bioconductor. So let's invite them up.
Good evening, everyone. So thank you so much for your patience. I apologize. We've had some technical difficulties, but we're going to pick up and we're going to do the best we can from here on out and still show you hopefully some really awesome things in Galaxy and Bioconductor. So my name is Jeremy Gex. I'm a longtime member of the Galaxy community. And tonight I'm going to act as your host for GBCC 2025 Live. It's my distinct pleasure to be able to introduce many people who are going to show you through various features of Galaxy and Bioconductor and also introduce you to some really exciting biology along the way. So as a community, and as we thought about this presentation, our goal was to try to piece together everything that's being done. Many of us are builders. And so as builders, let's see if I can start this video. As builders, we oftentimes focus on our small little piece of the puzzle. And we know those pieces come together into something bigger, but we don't often get to see that realization. So many of us are maybe builders of Bioconductor, builders of Galaxy, builders of software packages or tools or visualizations that go into these platforms. But seeing it all come together from a user's perspective and the power that comes from putting these things together, the sum is really truly greater than just the parts. And so that's what we want to try to demonstrate tonight. So what you're seeing here in this video is a very sped up version of how you build a car, of course. And so the analogy here is that all of us potentially contribute to these cars, or in our cases, these software platforms that are helping to change the world of bioinformatics. But sometimes you don't want to build, and instead what you'd prefer to do is drive. And so here, what we want to do is walk you through some of the most exciting features of Galaxy and Bioconductor that have come out over the last year or two and how these platforms can begin to work together. So one of the goals of this conference is really to cross-pollinate and say, what can Galaxy learn from Bioconductor and what can Bioconductor learn from Galaxy? And maybe we can build something better and bigger by putting our platforms more tightly together. You've already seen some examples of how this is already happening. You'll see tonight how Bioconductor has been integrated into Galaxy and how Galaxy has leveraged some of the same R scripts and packages from Bioconductor to make analyses easier. So with that said, let's drive using our vehicles for the night, Galaxy and Bioconductor. Now, as responsible drivers, though, we are going to start simple and move to the more complex stuff. So that's how the presentation is oriented tonight, starting off with some simple analyses, moving to more complex analyses, and eventually the full-fledged, potentially partially autonomous analyses. So the biology that I want to share with you tonight and that we'll be talking throughout the presentation tonight is spatial biology. And so for those of you that have been around long enough, you'll know that next generation sequencing has been around for 15, 20 years at this point. Some people still call it next generation sequencing. But when you think about what the truly novel biology is, or the novel assays that are out there these days, one of them is definitely spatial biology. And so with spatial biology, the analogy, and this is a nice one that I did not come up with, but I've appropriately used for tonight, is that our bulk sequencing from 10 years ago was we take a tissue, we grind it up, we figure out what's in there. It's kind of like a smoothie. Single cell takes us a step further though, and says that we can actually find those individual cells and know what they are. Well, spatial biology allows us not only to find these cells that are out there, but to look at the structure and the organization of tissue. This has proven to be incredibly important. And one way that you can see the evidence of this importance is that Nature Method's 2024 method of the year was spatial proteomics, the idea that you could get single cell spatial proteomic data out of tissues. I did a quick search the other day and looked at what spatial proteomics has been applied to. Here are three nice examples, really diverse examples across biomedical research: neurodegenerative diseases at the top, cancer in the middle, and COVID-19 at the bottom, all in service or all able to be investigated by using spatial proteomics. And so this is the point in the presentation where I get to make my first handoff to Drs. Allison Creasy and Dr. Enes Afghan, who are going to come up and show how you can start to, well, first they're going to explain some of the challenges around these spatial biology and spatial proteomics data sets, and then show us how you can analyze them using Galaxy and Bioconductor.
I'm mic'd up. Thank you, Jeremy. And I'll need Enes to help drive for me. So just to expand upon what Jeremy mentioned here, I want to give you a bit of an idea of what spatial proteomics is for anyone that's maybe not familiar with it. So within spatial proteomics, this is a really powerful approach within spatial biology where we can measure proteins, both their abundance and their precise location within a tissue at the same time. And we can measure multiple of them at the same time. And this becomes really powerful so that we can capture the state and the type of cells that are within our tissue. So I have up here some examples of what some of those images are. This is kind of the raw data set. And then what you can see on the right-hand side is what that looks like when we build these high-resolution spatial maps of what our cells and our tissue look like, as well as the types of cells that are there. And so for today, for our demo today, we're going to highlight some of the tools and software applications that both come from the Galaxy and Bioconductor communities and how they can be used to support spatial biology analyses such as spatial proteomics. And for today, we're going to focus on human breast cancers as our example. So within this experiment, we have 4 samples. There's 4 distinct subtypes of breast cancer, as well as 1 normal jejunum sample that comes from the small intestine. And you can see those original images there on the left. And these data are really powerful because what we can do with this by profiling these individual proteins as well as their location, we can ask really meaningful questions about the differences we see across these tissues, such as, are there immune populations that are differentially represented across our different breast cancer subtypes? Or do we see that there's distinct spatial patterns or organization across our cancerous versus non-cancerous tissue? But importantly, these data sets are very large and very complex, so they have really big data needs. They need to be able to efficiently process high-resolution images. We need to have and leverage tools and scalable data analysis pipelines and workflows. And because we're working with images as well as the downstream data sets, most users want to be able to actually interact with these data sets in a dynamic and visual context. All right, so that's okay. I just wanted to highlight on the left here some of the steps that go into actually processing these images, and they're quite computationally intensive. So Enes, how are we going to go about actually processing these data sets in Galaxy?
Over the past year, and it's come down to the last minutes to show that we can do this analysis together here now live. But because it is cutting edge, as we just learned, we're going to see how far we actually get. So let's all go live. So the way this is imagined is that I will drive from how do we go from data that Allison described from the method that Jeremy introduced and produce some interactive visual results right now, right here, hopefully in a matter of minutes. And so I'm going to navigate us through Galaxy and hand it off eventually to other pieces, from Bioconductor and all. And Allison is going to fill in as we get to some outputs and results with what, as a scientist, you may want to get after having run through this process. So with that, one thing that we have new as of this year, is brought up by Anton and Mike just in the last hour, is this repository of vetted Galaxy workflows or pipelines. And these are not some random workflows that a person assembled in an afternoon, decided to share. These go through a peer review process. They are regularly tested and updated. And the goal of this is to get analysts, people using these, closer to where they want to be quicker. And so currently there's over 100 of these available that have gone through this process and are, like I said, continuously updating. Again, because we're dealing with imaging data, we will filter this down to the imaging category and see, we have a handful of these in the available library. So if we explore what one of these gives us, we have a brief description, we have some author information, versioning, an overview, and then input data sets and the type of input values that it receives, the type of processing that will take place as we run this pipeline. And every one of these that's published here has the same level of information. You also get this visual diagram that connects all the tools, processes that are used in the pipeline, so you can kind of quickly gauge, is this something along the lines of what I'm looking at? And so, Allison, given your expertise with the data and all, does this...
Yeah, so as I showed you on the slides, there's multiple steps that are very intensive to be able to process these images. And we can go from basically the raw images. One of the first things we need to do is perform segmentation to actually identify where our cells are within those images. And then we want to quantify what's the protein expression within those individual cells. But we also want to know what type of cell is it. So we might go about phenotyping our cell as well as doing downstream spatial analysis and visualization. So Enes, this looks like exactly what we want. What do we do next?
All right. So, all right, we have data. We have a potential workflow. We need infrastructure, right? Otherwise, we're always accustomed to having to install all these tools, to having to have our own either laptop or HPC that this can run on. Well, as we also heard in the introduction, Galaxy Project offers this network of free nationally run servers known as usegalaxy.star that let you just get access to those resources and those tools for free without ideally installing anything. So today we'll use the US-based UseGalaxy server at usegalaxy.org and just click run workflow. Right, and so we went from that sort of library where we browse workflows to a workflow run form that if we click through this visual representation of the workflow, it is that same workflow that we were just exploring on the other site here now in a Galaxy visualization representation. Similarly, that same help that we saw earlier, you can see now in the context of the workflow run form, so you can see what types of values you need to supply to be able to run this workflow. So with that, we need some input data.
So what if I have my data on something like Dropbox or somewhere else?
Another cool new feature of Galaxy that's emerged over the last year is not only ability to to add these in line with our workflow, but we can upload these data sets from a variety of these remote files or remote file sources. So there's a number of them, including public or predominantly focused on public data repositories, But what we've just added in the last several months is this ability to link your private repositories, whether it be an Amazon S3 bucket or your private Dropbox, right into your Galaxy workspace. And this remains private between you and this account, so it's not like all the users of the server will get this. I've already gone through the OAuth process to get the link up my Galaxy account. So here we can just browse that the Dropbox folder as you would on your computer or whatnot. We have the files in this directory, and we select the ones that are composing the particular input. So there is a little bit of I guess cleanup that we need to perform here to make sure that the files are named as we'll make them recognizable later, and then pay attention to what's going to happen next. A clue is already on the screen, and the first person that notices and calls it out, I'm buying you a beer after the event. So you may miss it. Pay attention, right? So I just click the start, and so technically what you would imagine is that data moving from Dropbox to your Galaxy account now. Did you see what just happened? Who said instantly uploaded? All right, I hope you drink beer. Come find me afterwards. That's 3.5 gigs, 830 megs per file, just instantly available in your account. And it's due to some hashtag comparisons on the back end, because these data sets already exist on this Galaxy server. And so we compare those without actually transferring the data. They just become instantly available. And so just a huge time saver, bandwidth saver that you can lean into. And now we can open up our help here to kind of explore the details of, but I've done this, so I'll just proceed with the kind of formulating the rest of the inputs as needed.
Yeah, so while Ennis is working on filling this out, I'll just explain briefly what he's doing right now is building what's called a collection. It's often considered like a list. And we're taking these image files, which are OME TIFF files. These are the standard that are used within the microscopy community. And in addition to our image files, there's also a handful of other inputs we need to provide for this workflow to run. You can imagine that we might need to know what proteins were actually profiled within this experiment. We may need to also define how our cells are going to be phenotyped or labeled, and so we can come up with a strategy for doing that. And in some cases, you may also want to provide preset thresholds to say when is a protein actually present versus when are we just seeing some noise within the data.
All right, I'm nearly done. So we have these inputs selected, all right, so pay attention again. You may miss what's going to happen. And this is the beauty of a live demo, folks. So we went from browsing a static website with populated workflows to importing it into an existing infrastructure, getting some data from a private repository. We hit run in probably 5 minutes, right? And now these jobs are progressing quite quickly, if you ask me, through the workflow, right? So they go from gray when they have just been scheduled, to yellow when they're running, to ultimately green when they have successfully completed. And so where we were hung up just a few minutes ago was that so what's happening behind the scenes is that Galaxy is comparing your history runs or your past runs for the same input data sets and the same input parameters. And if those match, it will just mark the job as complete. So again, you don't have to wait for these jobs to complete every time. So if you have a workflow like this and you want to change some parameters at the very end of a workflow, you don't have to wait for the 2.5 hours or so that this workflow takes to run and complete to completion. However, this is a very beta feature. And so we are leaving this as an open line item to finalize during the hackathon at the end of the week. Hence the red box in the middle of the screen. And we will switch to a run that was in fact successful a week ago. Yeah. And so, yeah, this is what you would see when all the jobs complete. If you decided to run and wait 2.5 hours, they would in fact complete. And you are ultimately presented with some outputs. And I will let Allison tell you more about the results here.
All right, thank you, Ennis. So I just want to go through some of the outputs, because one of the really cool things we can do is first actually take a look at what the original data sets looked like that we provided to this workflow. So I'm going to look at some of the raw images here. Maybe we want to take a look at what was the normal jejunum sample from the small intestine. Hopefully I grabbed the right one. And this is opening up an image viewer called Avivator. And here we can see everything's backwards. We can see our original image. And so let's say that we want to know what different markers look like within this image. For example, within the jejunum, you're going to often find a lot of epithelial cells that are being labeled here, hopefully in blue in a second. We may also want to know how many immune cells are present, or if there's cytoskeletal components that are present. And I don't think any of these are loading, which is what I had actually thought would work. Just a little slow. And then we can also look at the smooth muscle. And so you can see that as an example with this normal tissue, we're seeing something that's very, very organized. And so I'll show you in a second then, when we look at our other outputs, what it looks like for our cancer tissues to be very disorganized. But one of the beauties of this workflow that we just run is not only can we see these individual inputs or outputs, but we can also see a collective report of all of the data sets and the outputs from the analyses that we performed. So here we're showing a bar plot that's all 4 of those samples that we've previously talked about. I'll use the example here with what we're calling A3. This is one of the subtypes of breast cancer, where this is a triple negative breast cancer, which we know to be it's going to have a higher proportion of what are called our basal epithelial cells, and so those are represented in blue. So we can see that this result is what we're expecting when we compare a triple negative breast cancer to the rest of our samples. But we can also look at our the spatial coordinates of those individual cells within a given tissue and see where our immune cells are versus we were talking just now about those basal epithelial cells, which are spread all throughout this tissue as well. We can also do things like a spatial analysis, where we start asking for these different cell types that are present, how often are they directly interacting with each other from a spatial context. And then the last really neat thing I'm going to show you is a dashboard that has multiple of these data sets actually linked together. Sorry. So for this one, we can see that as we scroll in, our little linker on the side there is going to pan both to our heat maps as well as our bar plots and as well as our UMAP. We could even turn off what are called our cell masks. That's actually labeled and identified where the cells are and has them colored with the phenotypes that we presented. So this is also being a little slow, so apologies about that. All that to say, this is a really nice summary that provides both that interactive experience that a user wants, as well as tying together all of the outputs into one location so that they can do their own exploration and discovery within their data sets. That I'll hand it off.
Thank you. Excellent. This mic is on. Don't clap yet, but you have a quick question? Okay, sure. Why not? We're going totally off script tonight. Let me get you the microphone, though. Yep.
Hello, that was pretty cool. A demonstration that didn't work as intended. But One of the challenges for all these spatial analysis is that, especially in the beginning, when you haven't trained the image, like it struggles with like segmentation and like learning which boundaries, what cells are. But it seems like this automated workflow seems to bypass all of these. How can you add a step where I can actually do some manual checkups?
Where you can do what? Sorry, I didn't hear.
Like, can I see if these cells are like properly segmented, for example? It's like you haven't
Yeah.
manually given any instructions on what are its boundaries.
So the way we actually provided that within this data set is we give a single marker to say this is the marker we want to use to define that boundary. The model we're using is an all-purpose large model, not foundation, but called Mesmer. And so it's all-purpose, and it does a pretty good job of actually capturing both nuclear as well as whole cell segmentation, as long as you have markers that are representative within your data set to do that. There's the benefit of using something like Galaxy is you can tie in any model for segmentation that you want. You could do that into this workflow, modify it yourself, create your own report, and you'd still get to then explore what those results look like with a modified workflow. So if you want to use cell pose, as long as you have the same markers, we have not needed to normalize our segmentation steps. However, quantifying the protein expression definitely would benefit from normalization. One of the benefits of the sample that we were showing today is this actually has multiple samples on the same image. And so you actually get a lot of good representation of the heterogeneity across samples, and you could use that as a background then to normalize to your other samples. Yeah.
Okay. Thank you, Allison. Thank you, Ennis. So as we kind of step forward in this analysis, the next question that I would ask is, we saw this beautiful example of being able to run through a workflow, but as this question that was just posed to the group kind of accentuates, sometimes you want interactive analyses to be able to explore your data in more depth and do custom things. And so that's where I think we want to invite Vince back up to talk about some work that he's been doing to support interactive analyses of these spatial proteomics data sets.
All right. While Vince is setting up, and I'll just get us ready for the next state of this. So as we said, Galaxy offers a lot of this compute capacity for free, managed software. And one category of tools that are available are known as interactive tools. These are complex applications, RStudio being a primary example that you can launch and interact with from within here. And you can send data back and forth between to Galaxy and RStudio. So we have an RStudio interactive tool here. And some of these tools, some of these outputs that we saw earlier, we can just select from our input dropdown here and hit run. And in a few seconds, we'll have our own sort of private version of RStudio that will have ingested the data that, again, in this context, was a part of that workflow. And then going to what Jeremy said and what we'll hear from Vince, you can then interact with that data within RStudio in a more sort of art environment and interactive world. So this process takes a handful of seconds. I think we're hopefully there. Nope, not quite yet. There it goes. And with that, I'll leave it to you, Vince.
All right, wonderful. Well, thank you. This has been really eye-opening for me. I'm almost sorry that RStudio is here outside of the context of the Galaxy sequences, the history components, all the other things you can work with. This is RStudio running on its own after, interacting here. And the key point, thanks to Ennis, is that we have generated in the workflow and in the Galaxy computing environment these H5AD files, which correspond to the annotated and quantified images, right? The images aren't there. These are the numerical representations of the proteomic assay applied to these images. And so the question is, in the presence of that data, how can I do some more work with R and Bioconductor in order to learn more? So let's use a package. I wrote the package, and I'm going to tell you a little bit about it while it's loading up. And this is going off script a little bit, but we'll load that up. It takes a moment or two. I'm just going to go to my own website to tell you a little more about it before we use it, because I don't really want to spend that much time waiting around for these computations. It's called GBCC.mcm. So it's for this conference, and it's working on, MC micro proteomics. I don't know if we've even mentioned that as the platform yet. And someone in here will know where this picture came from. In fact, articles, overview. Yes, that's right. That's what I wanted to show. Okay, so this is work of Lambda Moses, the spatial feature experiment, data structure, data model for this type of data. And what I decided to do was to use Bioconductor in conjunction with RStudio and Galaxy to ingest the MC Micro H5ADs that were produced in the workflow, bring them into this framework so that we can do some more spatial statistics that are not yet part of the workflow. That's what we're going to do. So going back to RStudio, what I've got here is my library is loaded up. And what I'm going to do now is find the file A3. That's the triple negative, if I remember correctly. So we'll call it TN equals, or let's say TN path equals dir, doing a little bit of R here, Galaxy inputs, right? Full equals true. And I'm just going to be a little nerdy here. There we go. So now we've got this path to this H5AD file, and I'm just going to run my function. Well, what is that function? Process underscore MC micro. And I think it's worth looking at this because we're going to scale this later. So the idea is I have a package in Bioconductor called ZEL-converter, which can take an H5AD and bring it in as a spatial experiment. And then I will convert that spatial experiment to a spatial feature experiment because it's easier to do more sophisticated statistics in that format. And then I will run a couple of these statistical algorithms that are very easy to run in spatial feature experiment or in the Voyager package, really. Run univariate and find spatial neighbors, et cetera. So this code is a transformation from H5AD into a spatial feature experiment with some added statistics that we're ultimately going to visualize. So let's run it. We'll call it TN equals process MC micro of TNPA. Okay? And this is going to take a couple of minutes. Well, maybe a minute or so. ZEL-converter now is actually doing a Python to R interop, going in and dealing with the H5AD, brought it into the spatial experiment, then I converted it to a spatial feature experiment, and now we're doing find spatial neighbors. We're producing a Moran plot, and this is the statistic I'm going to be looking at more, the local G statistics, and we'll visualize those in an app in just a moment. Questions or comments? Yes. Well, really, I've just been watching. I wrote a little bit of code to get this to go, but really what this is a way of thinking about a workflow that you can then sort of, as we discussed before, inject different types of modules to keep the workflow going, and then interrupt somewhere and find some of the data and do some interactive things with it. So a really wonderful experience for me, because I have never really done this before just a couple of weeks ago, looking at Galaxy. So here we are. Now, those of you who enjoy Shiny will be able to enjoy this. So I'm going to say browse one, because I didn't bother to browse them all, and I'll just put TN here. That's my new object. And here we go. We are going to be able to see the statistics, the get us or GI star, which is a measure of hot spotness or cold spotness in the spatial context for different markers here, which we can pick. So let's say I'm not too interested in RNA Pol2, but I'd like to see maybe CK5. I click that one off. I put in CK5, and now I get a new picture showing that there are hotspots of CK5 in certain local regions of the tissue. Well, that's all very nice. It's very, very concise code for doing this. You can get a picture of what the neighborhood structure is here. If you want to use a different approach to characterizing neighborhoods of cells, you have that. I'm not going to get into that. I'm going to finish up very quickly. Let's start this focused part just so you can see that. This will take a minute to compute. But what is really nice, I think, here is that the statistical tables, that take for every cell, give you all those GI star statistics. They're all here for every one of the markers. So these are tables of maybe 15,000 cells times 27 markers. They're all in there. They're not being shown because the focus is probably eating up all the CPU. But this is all part of this package. So you can use it yourself if you would like. Now, this is a plotly view of a certain subregion. And if you wanted to zoom in on something, let's say these guys over here, that's all easily done. And then you can see both the geometries of the cells that were called as we got to the H5AD. So you can see the ellipses. I calculated them in the program. And then the positions of the centers of the cells. So that's all there. And let's see if the tables ever came up. Yes. So any one of the markers that I want to see, all of the cell profiles, the get us word statistics, that's all in there. Okay? I think that's it. All right. Back to you.
Thank you, Vince. That was really interesting. This, I think, illustrates nicely how Bioconductor complements Galaxy, where Galaxy can do some of the more structured analyses, and then you come in with Bioconductor to dig deeper. Now, we want to loop backwards at this point and say, well, we have these nice Bioconductor analyses. How can we transition this back into Galaxy and move it into our more structured environment and potentially scale it up? So with that, let me introduce Marius van de Beek.
Yep. Thank you. So we're just going to do that. So we've seen the processing script that the process micro that it's just showed us. And we can take this and turn it back into a Galaxy tool. And I mean, this is a beta feature, and like we're testing this now, and hopefully around the next release, we make it somewhat public so that if you have like a trusted email, we would allow you to come and bring your own code directly into Galaxy without having to talk to one of us first, because that is currently how it is. Like you can write a tool, you can publish it, but it's a bit of a long, lengthy process. And the reason why that is, I will talk about tomorrow in my talk. But yeah, let's dive right into it. So we have this new little thing here, custom tools, and as I mentioned, hopefully soon we can make that available to the world. And then we're going to write a quick little Galaxy tool. And this is in fact not, for those who know Galaxy tools, this is not your typical Galaxy tool. It may look familiar in terms of what you're seeing here, but really this is a YAML tool, and there are some key differences we're going to go through. So I'm going to call this demo GBCC. And due to the technical problems we had earlier, this is almost certainly not going to work. But yeah. So we're just going to add a little description here. And then, you know what? I don't remember which container we have to use. So I'm going to go back to the account that we were planning to use. So let's pretend that didn't really happen. So I've already pre-written it here, but that's actually an interesting case in a way, because I'm just going to steal the code from here that I know works. Okay, so I mean, this is going to convert the H5AD that we use as input in the RStudio tool. I'm going to make a CSV out of it. Yeah, so I mean, this is the same container image that was used in the RStudio demo, but eventually you'll be able to bring any of the bio containers, and that's all going to be configurable. And then one of the key differences is, so in order to have a Galaxy tool, you need to describe the inputs so that the user can select them, and you need to describe the outputs so that they can be picked up and put into the history. And then finally, you need something to run, so that's our shell command. What we're doing here is we're going to symlink we're creating a symlink, and then here's a JavaScript expression with inputs corresponding to what we defined here. So we have a script input, we have an input 1 here. So we're symlinking this input 1 that is supposed to be an H5AD file, syncing this to input.h5AD, and then we're running the script. And the script is yeah, that's our script that we want to run. Yeah, so we just have to upload this script to our history. So we can do that right here. Right, so just to repeat, like we are symlinking the input, and then we are, the input is input.h5ad. That's because yeah, the script reads from this file called input.h5ad, and it's going to output back into spatial expression data.csv. And this is what we're picking up here from the working directory. Yeah, so you see here we have no tool. Once I hit save here, the tool appears here. And then you can either continue to edit it, or if you click on it, it just opens up the tool interface. We need to make sure to select the script and a collection of H5AD files. So this is the same input that Vince was using earlier. And then when we hit run, yeah, it starts running this tool. Yeah, and hopefully in a bit we'll see the output complete. There is some issues in getting this to run fast. So yeah. So maybe what we can do in the meantime is continue.
Yeah, absolutely. So let me just introduce and say that I know we're running up at 9 o'clock. You don't want to cut into your socialization / beer / downtime too much, but give us about 10 more minutes and we'll wrap this up, hopefully with some really cool things at the end. Marius, I think what you want to continue on to at this point is talking about how, if you want to run large-scale analyses using the tools of Galaxy and Bioconductor, but you want to work from the command line rather than the user interface, because maybe you're more of a bioinformatician or you have particular constraints that you're working within.
Yeah. Maybe your data is on an institutional HPC or some protected data that you can't easily move out, or yeah, maybe you just have a ton of resources there that we can provide to every single user of usegalaxy.org. So we already saw previously the IWC website, so we're going to pick up another workflow here. This time, another one for tissue microarray analysis, but this time slightly different. And all of these workflows also have a how to run tab. So here you can sort of pick, do you want to run in Galaxy? That's what we've done before. Or you can run with the Planemo command line interface. And in the interest of time, I'm going to skip installing Planemo. And I just want to point out that all the instructions are here. They should get you from the, not having run a Galaxy workflow before to getting it run on the command line. Importantly, for each workflow, there is a sample job config file. So this is where you put your inputs. And then there is a command that you want to run on your system. So let's switch to the terminal. And we're going to run this workflow with data that is already on the HPC. We're going to download the data after the run is complete. And we're going to create an export of this workflow run that we can then re-import into any Galaxy instance. This all took perhaps a little bit too long, so let's hope we're getting an allocation quickly. So yeah, I mean that's what you do. So you will log into the cluster head node, and now I'm requesting a job so that I'm not occupying the head node. That usually doesn't take too long, and I had this all prepared, but well, go from one technical problem to another. Oh yeah, and of course you could also there we are. Like you could also run this on your laptop, but we just got our job, so that's great. So I'm in my home directory, and all I have here, I mean, forget that there is a Miniforge folder. We're not going to use that. So in the demo folder, I already have some data. Let me clean that up. That's just from the previous test run. Okay. Let's see. Sort of checkpointed my work there. Okay. No, this is not very clean. Okay, it doesn't matter. We can get those back. Okay, so this is sort of the situation that you would be in if you followed the commands on the AWC website. And I have already pre-filled the job file, so it just contains the input data sets. These are not the same, but very similar files to what we used in the workflow on the command line. And just to illustrate, that's where the files actually live. And then we have the command here that we want to run. I mentioned we also want to create an export of the invocation. So just making sure that I'm this late in the process, not making any further mistakes. I already recorded the command. So what we're doing is running the workflow with this job file. I mean, that's fine. It'll not be in the same file that we were expecting. Anyway, I'm going to cheat later on. Yes, but thanks for coming to my stand-up show. No hecklers, that's good. All right, so I mean, basically what we're telling Planemo is run this workflow with this particular job definition. We want the outputs in this particular output directory, and then we want to export the invocation. And then what happens is that we've set up a throwaway instance of Galaxy, and then all the stuff that's needed. Look at that, I forgot. Profile. Chance to fix it. So this is the Anvil HPC, so I need to use the Anvil profile because that's going to set up everything that's needed to submit jobs against their Slurm system. So you can have all kinds of different profiles for the places you work at. And in fact, Planemo is sort of our command line interface for running workflows, for testing workflows, for running tools, for testing tools, for, yeah, I mean, generating these job files that I've shown earlier. Yeah, so it's currently setting up Galaxy, it's starting it up. This is a throwaway instance. We do that just for this particular moment. And then what it does is it starts Galaxy, it installs all the tools that are needed. And then it starts uploading the data. So that's what's happening now. And then in a moment, you should start seeing the workflow as it starts running. Yep, there it is. So there's a cool little progress bar. You see, yeah, that are jobs. So I mean, this is maybe not super readable, but here it says new. Here is like a, what's it called, a sandglass, hourglass, to indicate that it's waiting. And now 5 jobs should be running on the cluster. So can maybe in a different window connect and see what jobs are running under my account. Yeah, and there you see them. So these are the jobs that were launched by Planemo, basic illumination. I think this is this Ashlar? Okay, I'm not super familiar with the workflows. Anyway, so that's going to take like 10, 15 minutes. And of course, we're going to wait until they've completed. Or we take a shortcut and okay, so this is sort of what happens when like the workflow is complete. You get the invocation zip file, you get the outputs. So, I mean, if we look back at this, so this is the folder with the outputs, all the individual files that this workflow produces are there. And then we have the invocation file. And what we can do with the invocation file is, I'm just going to copy it quickly to my computer. And then we can import it in any Galaxy instance, whether that's a public one or a private one. So for instance, imagine that you want to share like this run with your colleague who's doing a demo and all the things he's trying failed. So then as a sort of backup, you could upload this workflow run that you did as a sort of backup mechanism on your local HPC and save the day. Yep, so it should finish pretty quickly. In the meantime, this is the tool we just wrote, and I mean, 3 out of 4 worked. That's not too bad, really. Why did it fail? Looks like a download error. Yeah, so I mean, this is setting up MiniForge as part of the script. That's, yeah, it can fail, but sometimes it works. And if we just did a simple cat or we ran something else that didn't need like a heavyweight setup process, it would have worked. So that's good. Yeah, and here's our new import. That's not the workflow we ran before. That's like on this machine. That's the workflow that was coming from Anvil. So again, you see the overview, you see the steps, you see the inputs, the outputs, everything is here. Yep. So some of the metadata is being regenerated upon import. Yep.
Fantastic. Thank you, Marius. Okay, you've seen some really fantastic stuff tonight as we've walked through this demo. Much of it worked, much of it's also a work in progress. We have one more thing to share with you. All good technology demos these days have some AI included in them. And so we saved you the best for last, where we're going to show you some AI that's been used to analyze our spatial biology data sets. So now Dan and Baker is going to walk us through that.
Well, if there was one part of this that wasn't going to go maybe exactly according to plan, this was the part that I was thinking was going to be it. So let's see where we get. Just for my own benefit here, I'm going to rename this just so we can live, just so we can find it. Okay, so I'm going to show you a completely different interface for Galaxy. Why didn't that update? I did. Oh, there you go. Okay. Okay, so this is a completely different interface for Galaxy that I think is pretty fun. It's called Claude. So if you're not familiar with Claude Desktop, it's ChatGPT. It's, you know, a standalone thing. This is the one from Anthropic. But they support these sort of interface layers called MCP servers. It basically allows Claude or your LLM, whatever it is, to talk to, I don't know, whatever you want, really, any kind of outside context. So I have pre-configured this with our Galaxy MCP. There's a talk tomorrow that'll go more in detail about it, but we just wanted to sort of show it off in the context of live. So I'm just going to start with, you know, we just did some work in Galaxy. Let's see if I can type any better than anyone else tonight, to ask a few questions about it. Can you connect to my Galaxy? Please. You are supposed to be polite to your LLM. I should have said please there. We'll see if it actually works. Okay, it likes me. So what's going on here is it's just a normal, chat interface, but it's actually using the tools that we've provided to it to connect to our Galaxy server. This is all sort of BioBlend under the hood. It's actually making API calls and talking to the real Galaxy server that hopefully we were just connected to. I see you're logged in as Marius. You're using whatever of your quota. I like to see that. That's great. Okay, so now let's say, awesome. Can you give me a short list of my recent histories, please? Okay. And we'll see if we can find the right history since we've sort of, played a little bit of the shell game here. So there, number 10, almost got cut off. Cool. Okay. That 10th one, GBCC TMA live, is the one I'm interested in. Can you give me a brief description of what we've done here? So now it should, oh God, I forgot to say please. It should grab that history, get a summary, pull down contents. This pulls it all into the LLM's context. So then it gets passed back to the model. It'll do some thinking for a second, and hopefully do a pretty decent job of summarizing what happened. Most of us probably know at least some of what just happened, but imagine, you're coming back 5 months later, you've done something. This could be a really nice sort of summarizing tool. Yeah, I'm going to assume that's right since I'm between you guys and getting out of here. So I like pictures. Pictures are a good way to visualize stuff. So can you make me a diagram that I will? Yes. Could you make me a diagram that I could share with folks? Maybe a mermaid diagram that I could work on, or okay, whatever. Share. Thanks. So it'll take all the summary information that it's pulled out of the history and the structure. You could also ask it to look at a workflow and summarize, but it's pulling it all in. This is Mermaid. If you haven't seen it before, it's a really nice sort of declarative way to make diagrams. And once it finishes, that should turn into a pretty picture. There, okay. We don't have to zoom into it now. And that's way less summarizing and more actual steps than I expected. But, one of the great things here is you could just iterate. You could ask it, okay, well, maybe I want 4 boxes. Can you summarize the different phases of our pipeline? That kind of thing. Again, we don't have to do it right now. And I don't know why I'm doing this in a tiny box in the middle of the screen instead of that. Okay, but so I guess the last thing I want to show is, at least in academia, right? If you didn't publish it, it never happened. So one of the things that I want to try is, the method section is always the worst section of the paper to write. You've got to track down all the details and synthesize it, and it's the least fun part of the paper to write. So let's see what Claude can do. Last thing we need to do tonight, or out of here, to draft methods section for publishing this analysis. I probably should have just copied and pasted all this. Let's grab the tool citations from Galaxy and draft that for me. Don't get to cite Galaxy and Bioconductor. Yes, I was already on it. Thank you. And this may take a while. It'll probably make a whole bunch of requests, which is good. That means I don't have to do it. But it'll crawl the Galaxy history, find all the tools you used, pull the citations if they're available from Galaxy. I don't have it enabled on this right now, but for citations that aren't available, if you enable web search, what this allows you to do is pull together all kinds of arbitrary resources in sort of one interface. And this is just Claude, but they're all the same. You can use this in ChatGPT Desktop or any of the other LLMs, right? But it's sort of a standard layer that we've exposed Galaxy to. And, it's going to do all this stuff. It's, finding all the tools. Oh, it's looking at the whole workflow invocation. Yep. So workflow structure, analysis pipelines, it pulled all the all the exact tool information. And it's going to write this up so we can head to the what's it called again? Campus bar. Yeah, but so this is incredibly powerful, right? This may not be the best example since we've sort of, seat of our pants here tonight, but I'm pretty happy with it.
Awesome. Anything
that's
else,
it.
Bannon? Nope. That's great. Thank you. Okay, real quickly, let me switch back to our slides and just wrap things up. So I'm just going to go to the acknowledgement slide. So this was a huge amount of work by a lot of people, not only the people you saw present tonight, but others as well. And so these individuals made particularly important contributions. Thank you so much, Allison, Ennis, Marius, Vince, and Dannon for presenting and doing a great job despite the technical challenges that we all encountered. Ennis was really the visionary behind this work, though, and thinking about how we put together this project. So he deserves an extra special thanks. Let's thank all our presenters one more time.